Hello, let's discuss a case today. So, a patient came to a doctor feeling congestion in lungs, spitting blood in his sputum, and he was also feeling very worried that his symptoms were overlapping with the symptoms shown in case of tuberculosis and lung tumor. But the doctor ordered a few tests, and what did they find? Let's see over here. They found these little things in his sputum. Now, what are these? These are the eggs of P. westermani or Paragonimus westermani, commonly called as Oriental lung fluke. Now, why is it called that? Let's look over here. Over here, you can see an organism which definitely does not look like a worm, right? Because worms are long. This organism is a fluke, which is classified under the phylum Platyhelminths. So these organisms are dorsoventrally flattened. Over here you can see the dimensions. Not very long, not very wide, but big enough to be observed with naked eye. They are oval in shape, as you can see, a bit roundish, reddish brown in color, much like a coffee bean. It has an oral sucker and a ventral sucker, which is pre-equatorial in position for the attachment to host body. Now this P. westermani or Paragonimus westermani is called as Oriental lung fluke because it inhabits the Oriental region of the world. Also, it is called as a lung fluke because it is present lodged in the lung of a human being. That's why we are studying it, because it is of humans' importance. Now let's look at its life cycle to understand what is going on. So remember the patient that we were talking about? A few days ago or a few months ago, he relished crabs. Now what is wrong with that? Well, he relished crabs that were not cooked. He was eating drunken crabs. Now you may be surprised that in parts of China, Korea, Japan, and even Northeast India, people enjoy crabs without cooking them and dipped in alcohol, called as drunken crabs. So that person enjoyed those drunken crabs which had this. What is this? This is a Metacercaria. This is a larval stage of P. westermani. You can imagine them as the dormant life stage of P. westermani. So over here you can see a sucker around the mouth, a mouth much like in case of adult, an acetabulum and a thick cyst wall. Over here also in the picture you can see a thick ball, and it has a gut also which is not terminating into an anus because platyhelminths have an incomplete digestive system, which compared to Taenia solium does not have a digestive system because of the fact that it does not need to digest anything. It is living inside the jejunum of a human being, so it is already getting the pre-digested food. Anyways, moving forward, so that person ate drunken crabs which had Metacercaria in it. So now the Metacercaria has entered his body, which exists in the duodenum. So the cyst wall, the thick cyst wall, it gets removed in the duodenum due to various secretions over here. Then it somehow migrates to the lungs where it gets lodged into this part. What is this? This is a granulomatous cyst. Over here also you can see a cyst, right? So a granulomatous cyst is generally the body's response. So when body sees that something is entering which is of foreign origin, it starts reacting. So granuloma, which are the cells and various other components of the body, they form a cyst around these flukes. So encapsulated organisms start ovipositioning in about 65 to 90 days. And remember in case of Taenia solium, which was as long as 1 meter, there was no chance of finding another Taenia solium. But in case of Paragonimus westermani, these organisms are not very big, right? So there is a chance that more than one organism is present inside the body of human, as in case of sheep liver fluke, where more than 200 liver flukes can be present at once in case of sheep. Anyways, moving forward, these organisms face each other's uterine pores so that cross fertilization can happen. Very smart. So in case of lung fluke, you can see cross fertilization happening. Once the fertilization happens, eggs are produced, which were found in the sputum of that patient. Now, these eggs are unembryonated when they are released. They can also be present in the feces when they are swallowed. And who swallows their sputum? Well, mostly everybody, but generally seen in case of kids. Anyways, so these unembryonated eggs get embryonated upon contact with oxygen, moisture and suitable temperature of 25 to 30 degrees Celsius. Now, it is very important for us to understand that the temperature 25 to 30 degrees Celsius is only present in tropical and subtropical regions. Hence, the presence of P. westermani in case of temperate regions, it's next to impossible. And note that moisture is very important in this case. Let's see why. So, upon finding these favorable conditions, the embryo inside the egg starts producing a meracidium. In about 2 to 3 weeks, it is already developed. Now, over here, you can see what a meracidium is. You can see cilia, you can see these plate-like things. So, cilia helps meracidium move. Now, in case of P. westermani over here, you did not see any cilia. Neither was a tail present. But in case of meracidium, we have cilia. So we can see that the cilia are present for distribution of Paragonimus westermani. And these eggs, how does meracidium hatch out of it? Well, in case of P. westermani, you will see a very peculiar thing. These eggs are operculated. What are operculated eggs? You can see the operculum, or you can visualize the operculum as a lid to a container. So when the meracidium has developed in 2 to 3 weeks, it just exits out of this operculum, this thing over here. Over here you can see a few characteristics of egg. So egg hatches into a meracidium. It is about this in dimension, 83 into 53 micrometer. It is yellow to dark brown in color. It is oval or elongated, right? And it is somewhat flattened in base. It has a very small operculum and it is thickened over here, right? It has a shell also. Over here you can see. Anyways, moving forward with the life cycle. So now meracidium has hatched and meracidium looks for its first intermediate host. Now the thing about flukes and tapeworms is that they are obligatory parasite. You cannot find these organisms chilling about in nature because they just are not capable of that. So meracidium, once it hatches out, it has to find its first intermediate host, which is a snail, a fresh water snail, in about 6 to 8 hours or it dies. Over here you can see cilia, dermal plates for its protection. It also has eyes. Not like eyes in case of humans, but these eyes are capable of photoreception so that it can find its host. So meracidium hatches out and has to find a host in about 6 to 8 hours or it dies. Its first intermediate hosts are freshwater snails of following genre: Thiara, Semisulcospira or Melanoides. Now let's assume that it did find its host, and after entering the snail, the freshwater snail entering through the soft tissue of snail, it starts losing its cilia and transforms into a sporocyst in about 1 to 3 days. So now Miracidium has transformed into a sporocyst. Over here you can see that sporocyst is a bit elongated in structure. It has germ balls. And what are these germ balls are for? Well, these germ balls produce radia asexually. Radia is the next stage of Paragonimus westermani. So this radia larva produces more radia in favorable conditions and it produces Cercaria in unfavorable condition. So radia produces more radia in favorable conditions, but radia produces Cercaria in unfavorable condition. Now why is that? Well, because an organism wants to multiply more and more in case of favorable condition when it has plenty food, plenty space, plenty everything. But in case of unfavorable conditions, it wants to further its life cycle, right? It wants to reproduce. So that's why it produces Cercaria. Even in case of small organisms like unicellular organisms like amoeba, you will see that once the unfavorable conditions set in, it forms a thick cyst wall around itself and starts reproducing, starts producing multiple daughter cells, which is different from its natural course of binary fission. Anyways, so let's just say that Cercaria has formed. Over here you can see what a Cercaria is. Cercaria is this elongated organism, but wait, it has this. What is this? This is a small tail. Now this is not a big tail so that it can swim around. It is a very small tail. Hence, this Cercaria of Paragonimus westermani is of microcircus type. So it just inches about on vegetation so that it finds its secondary intermediate host. It does not swim. As in case of sheep liver fluke, it inches around. Anyways, so Cercaria, let's just say it finds its secondary intermediate host, which is a crab which the patient ate, or a crayfish. Over here you can see that a crab is there and a crayfish is there. Crab of genus Potamiscus and crayfish of superfamily Stockoidea. So Cercaria has entered the crab or crayfish now. Now it has changed to Metacercaria. It has lost its tail, which is a very small tail, and it has lodged itself in the skeletal muscles of crab or crayfish. So over here we have already discussed the structure of Metacercaria, which is the next stage to Cercaria, right? So now Cercaria is changed into Metacercaria and Metacercaria is eaten by a human, which was showing the following symptoms that we had discussed earlier. The persistent cough, the blood in the cough, and the congestion in lung. That was because of the fact that a granulomatous cyst had formed, which was putting pressure on lungs. Anyways, it was the life cycle. Human ate crab, which had Metacercaria. Metacercaria changed into the adult. It started reproducing. Unembryonated operculated eggs were formed, which were embryonated upon finding right conditions of oxygen, moisture, and suitable temperature. Miracidium hatched out of it, changed into sporocyst, then radia, Cercaria, finally Metacercaria. Let's look at the hosts of P. westermani now. So in summary, we have a human, which is the definitive host because sexual reproduction takes place in human beings. Unembryonated eggs are found in the sputum of human beings. Also, in place of humans, you can also see sometimes that the members of Canidae and Felidae family are also infected with P. westermani, especially dogs, maybe even leopards. Anyways, the first intermediate host is a freshwater snail of Thiara, Semisulcospira or Melanoides genre. Melanoides is also a burrower, and all of these snails, these utilize both lentic as well as low-tech systems, which are still and running ecosystems, running water bodies. Crabs of Elcomon and Potamiscus genre, whereas crayfish of Stockoidea and Parastockoidea superfamily. So that was about it for the life cycle and a few of the symptoms that the Paragonimus westermani infection shows. Anyways, thank you so much for watching. I hope I see you again.